the slides. You can always find the slides and the materials available in the workshop in the Google Drive, and all our recordings will be available on the YouTube channel under the name of Multi-Omics Data Integration in Human Exposome Studies. Now we want to take advantage because, as I said, this is a very large group of people, and we have an open position at ISGlobal where we are looking for a postdoc researcher in exposome research. So if you are interested, or if you know someone who might be interested, please go to the ISGlobal website where you can find this job opening. Before we continue with the next session, just a small summary of what we learned in session 4 on the previous summary, where Jesse from the University of South California, he talked about, he showed to us a framework for evaluating the mediation role of multiple omics layers in environmental epidemiology. This cartoon, this figure here, it's actually a very nice summary figure of what Jesse presented during his talk. And the main conclusions of this multi-omic or high-dimensional mediation frameworks is that different approach for multi-omics integration can provide complementary insights on the environmental drivers. For instance, we have the high-dimensional mediation framework, where if we combine this by using different omics through inference on joint models, if we combine this by using with early integration, we can account for correlation between all features. Another approach that Jesse showcased was the mediation with latent factors, and this is particularly useful when we're exploring or when we are trying to understand distinct biological pathways. And also there is this integrated quasi-mediation approach, which is very important for identifying high-risk subgroups of the population. As I said, you can find all the material online in the Google Drive and see our talks on the YouTube channel. Proceeding now with, this is an overview of the work series. Now we are in session 5, where Léa will give us some insights on interpretation based on external knowledge. Léa, thank you very much for organizing this very interesting workshop along with Augusto. Just a brief overview of her Léa's bio. She's an assistant research professor and director of the Exposome Hub in ISGlobal. Léa has experience in numerous exposome-related projects, such as the Helix project, the Athlete project, and the Endomic projects. And her main lines of research include studying of the exposome in pregnancy, in childhood and adolescence. She is focused on metabolomics data integration in epidemiological studies and how this can relate with external stressor. And she's particularly interested in neurodevelopment and mental health. So at this point, I will stop sharing, and then Léa, the floor is yours.
Thank you very much, Nikos, for helping sharing this workshop. So today's session, I'm going to focus on network visualization. And please bear with me. It's the first time I'm going to explain this more theoretically and more in details. So please don't hesitate to write in the chat if you have any question. And I'm going to make pauses, like switch between slides and demos. So I hope you will be dynamic and understandable. So I'm going to share my slide. Okay.
We can see it.
Yeah.
Perfectly.
Okay. I'm trying to switch my slides. Okay, so I'm going to start with introducing why, what are we trying to do with multi-omic integration, and introduce the idea of a network, and then go into a little bit of graph theory to understand how you can describe a network based on some where does network science come from and how we apply it in omics fields and exposome. And then I'll give you 3 case studies if I have time. In particular, how to interpret this network. I'm not going to speak a lot about all the statistical tools to build networks. I'm going to give some examples, but it's not going to be an exhaustive list of statistical tools. I'm going to focus more on how you describe a network once you have it. And there will be a hands-on component. I only have 15 minutes, so I'm not going to go step by step in detail, but I will share this. It's a Word document where I explain how to do it in a software called Cytoscape. So that will be shared with the people here. So network is really about how to describe complexity, and this is particularly relevant in biological systems. For example, in metabolism, you can describe a metabolic pathway in a very linear way from how glucose is metabolized down a specific metabolic pathway. But there's also a high degree of connectivity between glucose pathway and its metabolite with other pathways. And then you can also go into a more complex level on how the different genes are related to glucose pathway in different organs, etc. So really studying only individual part of metabolism, it's an example, but it's true for all the omics layer, is not really meaningful because we're not really measuring a single metabolic pathway. But if you look at an exposure impact on the metabolite, you're actually interested in a full metabolic pathway within a complete network. And one metabolite being affected by an exposure will have an impact on a larger network. And when we talk about omics, well, it's not the first session of the multi-omic of the workshop, but just to go back how these are measured, some are more related to nucleic acid sequencing based method, and others based on mass spectrometry. So actually, there is some challenge on how to integrate this omics data based on different nature. They're not all related down to a gene. And it has a very large scale. So here I'm giving an example of how many elements there are per omic layers, and if you add the exposome to that, we don't even know the size of the exposome. So there is an information overload, and if you start to actually look at the interaction and the association, then you have even more possible links, and it is very large to describe. So why do we need an omic data integration through a network view? Because it will help us to have a more comprehensive overview of the underlying signaling network and understand better understand the interaction. Also, if you start to combine the different omics, each omics have the drawback, the technical variability, or the coverage limitation. And by combining this omic layers, you can also maybe compensate for the noise inherent of the omic layer and have more reliable causal inference. one way to integrate this is to do pathway aggregation. So for example, if you have a metabolomic data set and a transcriptomic data set, you can look for metabolite enrichment and transcriptomic enrichment on the other side, and then go down to, for example, the KEGG pathway and look if these pathways enriched. And there are different ways to do this. I've put some tools here. Here I'm more interested in metabolomics, so I'm going to show an example of the metaboanalyst tool. So this is the tool offered there. You can put your list of genes that are related, for example, to a disease. And on the right side, you put your metabolites, which you need a KEGG ID, which that's difficult sometimes because you need a good annotation, and that's not always the case for metabolomic or exposomic, chemical exposomic. And then it would give you a pathway impact based on enrichment for certain elements. Okay, this is nice, but basically you need a lot of information about at the gene level, about the pathway level, and sometimes we are limited because of the limited biological knowledge we have, and it is even more true at the metabolite and exposure level. There's a lot of things that are not annotated, that are not biologically, functionally annotated. So this is why when we start to integrate the exposome, we also have to consider data-driven approaches to networks and not only biologically pathway analysis. So this is where I want to introduce the network science. So we want to embrace this complexity of the potential interaction across the omics, and network science is a great way to do this. It emerged in the past decades, and it's not only in the field of system biology, but it's basically in many other fields. Network has been very important to describe, for example, social network, to describe internet network, etc. And also at the brain level, like brain connection network. Here with the map, the first description of the full mouse brain network. So it's not specific to system biology, and there are strong forces, two forces that help the emergence of network science. It's the emergence of network maps, because to be able to describe the complexity and the networks, you need to have their connection and the exhaustive list of this connection. So the importance of maps. For example, with internet and website, we are able to we have the map of that allows us to build a network. And the other part is that there is a universality in the characteristic of these networks. And that's where mathematical models are very helpful, because they don't have to be specific to metabolic network or Facebook social network. All these networks, they have commonalities. And a lot of the concepts I'm going to describe now are very well explained in this book by Albert Barabasi. He's the main person who drove this field of network science. So now I'm not going to go, I don't have time to go in depth of graph theory. That's the mathematical field behind network science, but just to give you the key words to be able to describe networks. So what is a network? It's the catalog of a system components. And what are the components of a system? They're called nodes. So for example, when you look at a network of computer, each computer is a node. Or there is also, they like to do network for actors in movies. So each actor is a node or can be a protein. And then the link, the interaction are called links or edges. And networks can have different properties. They can be undirected, which means the association between the node 1 and node 2, for example, can be in both directions, or they can be directed. This is a case, for example, for website links. When you click on one link, it brings you to one website, but it's not the other way around. Okay. And then there are one main other characteristic of networks. It can be weighted or unweighted. So you can see at the bottom left, the weighted network, the links can be more or less strong. And so the link will not just be coded 0 or 1, but it can be on a different scale. And to describe a network, you need to describe it in a table as an adjacency matrix. Okay, and one of the main properties of networks in reality is that they are scale-free. So if a network was random, you would have possibility to have link between all the nodes, all the component. But actually in nature, that's not the case. Some nodes are more connected. So this is the case in the graph on the right side. This is the case, for example, if you think about social network, some people are much more connected, have more family, friends than others. And that's the case also for protein interaction, for metabolites. So this is a very important properties because that's what will help us to describe and infer biological properties. So I'm giving here another illustration of the human diseasome. So there are researchers that wanted to describe how the genes related to disease tend to interact with each other. So for example, I zoomed here on this cluster of disease, for example, hypertension, asthma, Alzheimer. These disease circles are bigger because they have more gene associated with the disease, and they also related because they have common genes associated. So here we can call them hubs. So hypertension, asthma, a disease hub based on the genetic association. Okay, some other aspect of network to describe is the centrality. So how important a node in a network is. And one of the most simple to understand, a simple characteristic is the degree. So if a node is associated with lots of other nodes, it would have a larger degree. It's basically the number of connection each node has. So in this drawing on the right, the yellow nodes have a higher degree than others. And then there are other derivatives of these characteristics. So a hub is a node with a high degree. Okay, I want to go now just to an example. So in the Helix project, we had information on many exposures from the external and internal environment in children and their pregnant mothers. And we were interested to look at what are the molecular signatures of the exposome. We looked at multi-omic layers from DNA methylation down to metabolites. So we had 6 different omics layers, and we looked systematically at all the association between each exposures and each omic. So we did basically regression models, and we tested 30 million associations. Out of this, using different significance threshold to filter the significant association, we ended up with a list of around 1,100 exposure-omic association. Then how to visualize this? So we had different questions. For example, is a molecular feature connected to several exposures, or are exposure connected to multiple molecular features? So here, the first result plot we had, it was to describe per exposure families. So here in this Miami, you have two Miami plots, and the different box of colors correspond to different exposure families. The top one corresponds to exposures measured during pregnancy, and the bottom plot corresponds to exposures measured during childhood. And each dot corresponds to one omic marker associated with one exposure. So this is very well. We could see, for example, in pregnancies, there are more blue dots, which were methylation sites associated with exposures. And at the bottom, we see more red dots, which represent more serum metabolites. But it doesn't really tell us, okay, are there more red dots at the bottom because there's one metabolic pathway more often associated with exposures, or are there different metabolites? So that's one of the questions. So to visualize and understand this result, we had to build a network. And we built two different networks, one for the pregnancy exposures and one for the childhood exposures. And you can see these networks look very different in pregnancy and childhood. In pregnancy, we have three main connected components. So there is very little connectivity. Whereas on the childhood exposome network, we see there's much more links between the exposures and the omics. So I'm going to show you how we actually talked about this in the paper. I'm trying to exit my screen. Okay. So to describe this, we talked about the average connection per node, so the average degree. And we could see, for example, in the pregnancy network, that on average, a node had only one connection, 1.3 connection. Whereas the childhood exposome network had an average of 1.9 connection. And we also had another type of descriptive, the shortest path length. So how two nodes were connected, how many nodes they had to go through to reach, to be linked, to be passed. So this was another way. We also described in the largest connected component how many nodes there were. So if you see here, in the childhood exposome, we had mainly one big connected component. That actually contained 90% of the associations. So to describe this, we actually had to do a further cluster analysis. Can we, in this big connected component, focus on more specific clusters? Are there nodes that are more tightly related that we could describe? So to do this, we had to use other mathematical models. So I'm going to go into Cytoscape to show you how we did this. Okay, so this is the software, Cytoscape, that we use to draw these networks. There are other ways that in R, there are ways to draw network, but I would say Cytoscape, the advantage, well, it's a free software, and there's a lot more flexibility, like how to draw the network, how to color it. For example, you can choose how big each nodes can be based on the degree, and this was very helpful to describe our results. And just to show you how we did this. So based on a table of all our results, so we basically had a table with a column for the exposure, a column for the omics features, and every possible connection with p-value and effect size. So we imported that to Cytoscape, and we asked in Cytoscape there is a network analyzer plugin that helps us to analyze our network. So it describes the number of unique nodes. As I said, in this paper, we found around 1,000 significant associations. Out of 1,000 associations, there were actually only 200 unique nodes, so unique exposure or omics features, and 500 ages. So this was quickly calculated from the Cytoscape. And then we can choose like different ways to show our omics. For example, if the serum metabolome, we put it in red or in yellow. So there's ways to play with that. And then we wanted to create sub-networks because basically when we look at this, especially the childhood network, it's very hard to see anything. So we wanted to be able to focus on sub-clusters. But how do you define these clusters? So to do this, we used this app in Cytoscape called Cluster Maker. So in app, Cluster Maker, Cluster Network, and we did a community cluster. And that added to our list where we had all the omics and like all the list of edges or links. It added a cluster number. So I forgot how many clusters we found, but like more than 10 clusters. And then we could select our clusters and plot them separately. So starting to create sub-networks. And then we could plot them separately. So I'll show you an example. So this is a full childhood network. And then, for example, we had a subcluster around organochlorine pesticides. So we could find that PCB 118, HCB, they had a lot of connection. But that was very helpful to see, okay, actually there's a lot of CPGs associated with this PCB, but some proteins are actually related to several exposures at a time. Or some metabolites were associated with several exposures at a time. And their size were related to the degree. We had also another network related here with several exposures, PFAS, heavy metals such as arsenic and mercury. So then the next question was, okay, we find all these omics associated with this exposure, so they must have something in common. How could we look for enrichment in these clusters? So for omics layers that could be linked to a gene, that's quite easy. There's a lot of established tools. But for metabolomic, it was a bit more tricky because although in that paper we had targeted metabolomic, still the annotation, for example, to lipids, etc., it's not so clear how to link it to a metabolic pathway. And also, maybe here what we were interested in is to look at potential sources of these exposures. Is there a common source to these exposures? And actually, I'm going to open the floor and ask you, what do you think is the common source of exposure to these chemicals? So these metabolic signatures in serum and urine, what do you think make this exposure related? What's the relationship? I'm going to open the chat. Okay, Sarah, she said fish. Very good. Yes, it is fish. But how do you systematically check this? You could check the literature on every single omic marker, or in these papers, they found that omega-3s are related to fish. Yeah, but we have many associations, and not just for these chemicals, but for many other chemicals. So we looked at it systematically to do metabolite enrichment. And one of the databases that's really good for this is Exposome-explorer. So it's this database that was developed by IARC, which is a WHO institution. And so actually this database, the origin of it was, I think, a phenol database. So the person developing was really into nutrition. But this database is then really extended. And they have catalogued with a manual curation all the publications that reported association with exposures of food biomarkers, and they have a lot of metabolite information in there. And they also recently added macrobial origin metabolites. So in our paper, what we did, we downloaded we downloaded different tables, the biomarkers, concentration, and metabolomic association, made it in one database, and look for enrichment in our clusters. So this is the result. So here was one of the clusters related to all these chemicals. We put them we put them together in a three-parted graph, and we found that these metabolites were actually related based on other publication to fish, fatty and non-fatty fish, for example. And it's not perfect, and actually in this paper we didn't have a full coverage of the metabolome. Actually in urine we only had 40 metabolites, in serum 180. But actually, but this paper is already quite old, and you could think now with the new metabolic and exposome platform to also use these tools to systematically enrich for dietary sources or macrobial sources. And one of the other clusters, it was related to some of the organophosphate pesticides. These are the gray diamond here. They had a lot of urine metabolite associated to them. And we found that this mainly represented fruit intake, based also on the Exposome-explorer enrichment. So we also did extensive enrichment based on the gene level, but I'm not going to show this because there are many other papers on how to do this. Here I put the catalog to the full results of these papers, and so you can actually practice between the catalog and the Word document I can share. You can practice how to do this type of network and enrichment. Okay. One main limitation of this paper, we didn't take into account the correlation within each omic layer. So we built a network only based on association between exposure and omic. But a different way to do this would have been to look at the within omic correlation and how these are related to correlation with the exposures. So there are statistical tools to do this. The most simple way to do Pearson correlation, but as you can imagine, we had 30 million association just looking at exposure omic. But here, if you start to look at within omic association, you have too many. So you need to apply sparsity to this correlation network. One way to do this is to use regression-based approaches, such as partial least square or elastic net. I'm just showing here, it's one example, and there are other ways to do this, it's many packages. But for example, if you use the R package in the Mix-Omic called sparse partial least square regression. It allows you to calculate, even on very large matrices of multi-omic data, to create this matrix that then allows you to build a network. And I'm going to show you two study cases where we did this type of analysis. The first study, it was in the panel study in Helix, where we had 2 time points of sampling, where we did also a multi-omic analysis. It was in 156 children, and they were visited at 6 months difference. And we did, we used this software called, the R package called GeneNet. And what we could look at, it's the omics covariability 6 months at 6 months of interval. And we had a lot more DNA methylation markers. So a lot more association between the DNA methylation sites, just because of the different size of the omic. But when we looked at connected components that had heterogeneous omic markers, we could find an interesting association. For example, the cluster D, where we had glucose measured and different CPGs that varied together. We found that some of these CPG sites were annotated to genes related to diabetes. So at least that made sense. Okay. And then another example is in this paper where we were interested at endocrine disrupting chemicals, how they were associated to multi-omic profile. So it was in the same Helix child panel where we had 156 children with the multi-omic measured twice. And we had also information on their chemical exposures measured in urine. So here we were interested, we used this repeated design to give more confidence to our network. So here I'm going straight to the details. So how we did that, it was a lot of interaction to test. So we had to use more sparsity. So we built a Gaussian graphical model using regularized pairwise partial correlation. There is a function for that, it's called peak or shrink. And we could also assign a p-value to this link. Then we did another pruning of this network based on significant association. And we did a last step of pruning of this network based on if this association were repeated across the 2 time points. And we obtained this network. And here's the difference with the first case study on the molecular signatures of the exposome is that we took into account the interaction within omics. So to show this, to summarize this network, we summarized how many edges or links we found between exposures. So here it's the first bar plot on the bottom left. How many links are there between exposures and proteins, between exposures and serum metabolites, exposure between urine metabolites? So this type of interaction were the least frequent, because as you can imagine, most interaction is going to be within omics layer. For example, between methylation markers or between metabolites. And this is how you can represent it. So lots of methylation interaction and serum metabolite interaction. One thing is to say that in this study, we were a bit smarter in how to deal with the different sizes of omics. For DNA methylation, we did a pre-processing step where we selected CPG sites that were associated with the chemical exposures. Because one problem we have with multi-omic integration with exposomic is that the association between environmental exposures and omics is much weaker than within omics interaction. So we somehow have to enrich this kind of interaction because that's what we're interested in. We're not interested in just representing the metabolomic network or methylation network. There are other studies for that. Okay, and one last step in this paper to do the biological enrichment was to focus on clusters that contain both chemical exposure and omic. And then we looked in the literature if this association were found in other type of models, whether in vitro, in vivo, and we would put more confidence into this association. Okay. So I finished my presentation. Well, these studies were built on a big collaboration network. This is what I wanted to acknowledge. I'm going to open the floor to questions. Maybe I'll stop sharing.
Léa, thank you very much for this very interesting presentation. And it is amazing, this work that you've done. I see now from the chat that we have 3 questions. Perhaps we can start with them and then give some times to the others to think of some. So the first question was about when you are showing the different networks and how they are developed. So Loreto asks if these networks, they don't show how strong the correlation is or how strong the connection is. Is this right?
So there is a way to show how strong the connection is. It's by doing a weighted network. I can show concretely how you do this. I think this is the point of this workshop. So for example, in Cytoscape, here is my network. And here I have information on, for example, I can put my beta coefficient or my correlation coefficient, depends how you build your network. And then I can use this information. I don't have it here, but I'll show it. I can, oops. When I build my cluster, I can choose, for example, if I do here, I can choose to use the weight of the network to determine the clusters, or I can even to draw it. Sorry. So just the layout, I can choose, yeah, edge weighted force directed layout. Then I can choose, okay, sorry, yeah, I was looking at. So here it's called log FC. That was basically our beta coefficient. So I do this, and then it will build a network based on the strength of association. So that's one choice to do. Yeah, I hope I answered the question.
Okay, thank you very much. Then we proceed to the next question by Mireia. And Mireia asks if the construction of these networks follow a known network topology, such as fitting a scale-free topology criterion.
Yes, so I'm going back to theory. In real life, not just biology, most networks are scale-free. So we are calculating and visualize the network on a scale-free topology. I don't know if she wants to ask the question. Yeah, if she wants to open her mic, maybe to ask the more detailed question about this.
Yeah, thank you, Léa, for the answer. Sorry for the question. Maybe it was not very precise, but I wanted to ask what was the threshold that you considered to be able to know that the network is following a scale-free network?
So I can answer the question on basically how what threshold you use to draw a link between two nodes. And that will depend on the three case studies I presented. We have different criteria, but from simple to complex. So in the first paper, the edges, we draw them based on the regression coefficient and multiple testing correction. So that's as basic, like as we do in epidemiology, in traditional studies. In other types of networks, like this one, it's a bit more complex. We use the series of rules. So we first use a regularized correlation. So it's similar to lasso. So you do first, you select based on the predictability of your link, and then we also filtered by p-value. And in that case, we also, because we had a repeated design, we also selected the edges that were reproducible. So I would say it depends a bit on your study, on the size of your network, and how much association you have. Because some, for example, the metabolome is very interconnected, so maybe you need to be more stringent. But if you're more interested in exposure-omics, then you want to be less stringent. So, you know, that answer your question.
Thank you.
Now we proceed to the next question by Adria, and Adria is asking, probably this refers to your example, Léa, where you asked us to guess what was the main source of a specific cluster within the network. So Adria is asking if the enrichment can be done with other potential sources other than diet, such as drinking water, breathing air, maternal burden, etc.
Yes, that's for sure. You just need the data. So here for the food enrichment and the metabolite enrichment, we could find the Exposome-explorer database, so it was easy to do. But for water, you just need to have a very good database that would have all the, I don't know, the water pollutant, or maybe, yeah, you just need another source of data.
And usually there are many external libraries nowadays where you can find this source of information.
Yeah.
Okay, let's proceed to the next question. Léa, we have Ismail asking, how do you connect different disease phenotypes or different clinical domains, such as cardiac, digestive, endocrine, pulmonary, etc.? So probably this refers to the disease some.
Yeah. So, and I'm just going to the paper. So in this paper, for example, where we looked at the molecular signatures of the exposome, we looked at different catalogs of enrichment, and we looked at if any of the omics markers, well, actually, okay, the first one we looked at epigenetic markers. So for this, for example, there is a very good catalog, the EWAS catalog, that contain all the previous epigenetic studies for exposure, disease association, clinical studies. So we looked at our epigenetic markers, how do they overlap with previous studies on, for example, liver disease, or you can read here, schizophrenia, and then that help us to enrich and better interpret our association. There is, for disease, there is also the David database, which maybe we didn't put it here, sorry, I forgot. But one limitation we had in this paper is that we were interested in, well, it's in children, and a lot of information in these databases about at the gene level or even epigenetic, etc. It's been studied in the framework of cancer, and we lack a lot of information of other type of clinical manifestation that are more relevant for children and general population. Yeah, a lot of omics, basically the omics field and system biology, it started in the field of cancer. And now we are applying it to exposome studies in general population in early life, and we are missing a bit of databases that would help us interpret our results.
So yeah, it is always important to bear in mind that these are examples of coming from children, which who are a relatively healthy population. So there we don't have these very hard outcomes.
Yeah. Yeah.
So we have another questions. We have a couple of questions, and I think we are still good in time. We still have 6 minutes. So the next question asks about if the same number of or if the features, if it should be the same number of features in each omics in order to build a multi-omics network. Because if there are different numbers of features, then you might get an overrepresentation of one specific omic layer.
Yes, exactly. So, for example, yeah, we had this problem with the epigenetic data where we have much more features than for metabolomic or proteins, and that's why we had to do a pre-selection of the epigenetic data, and that will depend on your study aim. In the case of this study, we were particularly interested in association with endocrine disruptor chemicals. So we pre-selected CPGs that were associated with endocrine disruptors using a quite relaxed threshold, but then that helped us to reduce the data set. It doesn't need to be exactly the same number of omics features, but I would say a similar scale. Yeah. Well, in that case, we had 500,000 CPGs and 40 proteins. So you can imagine. But,
okay, then to the next, we have 2 more questions. Léa, if have you used ever this type of networks with time consideration, and perhaps an example is with Markov theory.
With what concentration, sorry?
With time consideration, by considering time. I don't know, perhaps Etienne wants to open his or her mic to specify more the question.
Yeah, thank you. Yeah, I'm wondering if you can consider at different time the same people and see if there is different concentration and see the evolution of the disease with that, with some graph theory, but with considering different time.
Yeah, it would depend what's your question. For us here, time, we were interested in if we could observe the same association over time. We used it as a reproducibility. But maybe your question is different. Maybe it's opposite. Maybe you want to ask what's the difference in my network across time points?
Yeah.
Like, for example,
exactly.
pregnancy, I'm interested in if exposure are differently associated with omics network in the first and third trimester, then you can do a differential network analysis. So yeah, it depends on your research question. I just put a slide here. In terms of how to calculate the network, there is, for example, PLS that has a multi-level option that accounts for repeated measurements. But you just need to be aware, okay, how do you want to take time into account? What's your question? Actually, sorry, and one other example, in the case study 2, we also had repeated time. And there our question was actually how do the omics co-vary? So we were exclusively interested in the difference. So we subtracted each omic. So for example, glucose, we looked at the difference between the 2 time points and modeled that in the network. So yeah, it depends on the question.
Okay, thank you. So it's more for retry than to follow the same people and see the okay. Thank you.
Okay, and we go for the last question of this session. It's about metabolomics, and Lucia is asking where the high correlation between the features are common. What strategies can be employed in order to minimize the risk of type 2 error when applying more stringent criteria for statistical analysis?
Yeah. Well, I think that's the nice part about doing network is that, for example, here in this example where we had all these lipid-related metabolites associated with fish intake, maybe the point here is not to select the one metabolite most associated, is to have a good representation of the metabolic pathway. You don't need to select one, but you want to be able to consider them together. And that's what cluster analysis allowed us to do, not to interpret one by one, but interpret it together. And also you can do pathway enrichments if there is annotated pathways. So yeah, maybe you don't need to be super stringent, you just need to interpret it together.
Okay, and we are just on time. Léa, thank you very much for this great presentation. It was very helpful. Thank you all of you for attending this workshop. And yeah, we hope to see you in the next time. Thank you all very much.
Thank you.